Overview of methods and concepts in parasitology. Some detection methods for parasitic infections include microscopic examination. So this would be the identification of parasites or their ovocysts, which are eggs, in clinical specimens. We can also do cultivation, which would be growing certain parasites in specialized media or cell cultures for identification. We can also do immunodiagnostic or serologic methods. So this would be the detection of host antibodies against the parasitic antigen, which would be useful for diagnosing infections where parasites are actually difficult to find directly. The helminths, so these are parasitic worms that include nematodes or roundworms, trematodes or flukes, and cestodes, which are tapeworms. So we have an ecological relationship for these. So we have commensalism. This is where one organism benefits while the other, usually the host, neither benefits nor is harmed. And we also have parasitism, where one organism, which is the parasite, benefits at the expense of the host, which can cause harm or disease. Some parasitic host definitions. We have accidental host. This is a host species that is not the usual or natural host for the parasite. Parasite infects but may not complete its life cycle or causes atypical disease. Then we have zoonosis, which is a disease that is primarily of animals but can be transmitted to humans. Humans are often the accidental host. Then we have the definitive host. This is where the host in which the parasite reaches sexual maturity and undergoes reproduction. Intermediate host, so this is the host that harbors the immature or larval stages of the parasite. This is essential for parasite development, but not the site of sexual reproduction. With transmission of parasites, there are three elements for transmission. So you have the source of infection, a mode of transmission, and a susceptible host. The source of infection may be a cyst or trophozoite form of the protozoan. We'll talk more about that as we get into each of the parasites that we'll discuss. Transmission can also be through inhalation or ingestion of the eggs or infective larvae that are the source of those infections. So other ways of transmission are ingestion of contaminated food or water, some type of direct contact, injection of the parasite by an arthropod vector such as Plasmodium, which causes malaria. So that, the mosquito or the insect is the vector that carries those parasites, will then penetrate the skin. There's also an example of transplacental infection and respiratory infections as well. Other methods include poor hygiene of food handlers, feces that's contaminated in the water. So that would be like a fecal-oral route or inhalation such as pinworm. And then there is also sexual contact, and that would be related to an organism called Trichomonas. Some common specimens, we would get stool. So this is the most commonly used specimen for detecting intestinal parasites. We can do cerebrospinal fluid, which we would examine within 20 minutes of collection for motile forms. And sputum, so this would be used to detect lung parasites. We could also have genital urinary specimens, and we could also have urine. Specimen collection and processing for parasite examinations. Some lab methods for parasite detection is we have direct microscopic examination. This is the primary method for identifying parasites in clinical specimen. This can detect the eggs, the cyst, the trophozoites, the larvae, or the adult forms. It includes techniques like wet mounts, concentration methods, and staining methods. For cultivation, so growing parasites in specialized culture, media, or cell lines, this is used mainly for protozoa like Trichomonas, but it could allow for better identification and study of life cycle stages. Then we also have immunoserologic tests. So these detect host antibodies against parasitic antigens or parasite antigens themselves. This is useful for diagnosing infections where parasites are difficult to detect microscopically, and we can identify past or chronic infections. An example would be like an indirect immunofluorescence test or an ELISA test. With the parasitology in the microbiology lab, the microscope needs to include an ocular micrometer in one of the oculars. That will help with determining the size of the parasites, because that is a lot of times how we can make our determination on what we are looking at. So this is, like I mentioned, valuable information for determining the genus and species. Your centrifuge also needs to be a swinging bucket type rotor and not a fixed angled head rotor. And your centrifuge also should be explosion proof, and we'll discuss this in a little bit. So we have two major groups, specimens and methods for what we call lumen-dwelling parasites. So these are parasites that are found in the gastrointestinal tract, the pulmonary system, and the genital urinary tract. Lumen-dwelling via parasite is usually confirmed by a direct exam in either sputum or urine. And your method is specimen for blood and tissue. So those are going to be direct exam of a blood or tissue sample. We prefer that stool for ova and parasite, or we also can call it an O&P, is collected prior to no antimicrobial agents given to the patient, no barium, no mineral oil, no anti-diarrhea or laxatives given within the past 10 days. And sometimes with certain patient populations, it can be a challenge to make sure that they haven't been given any of those things. If at all possible, examine the stool within 30 minutes to an hour after collection. If the stool consistency is soft or liquid, we need to be looking for the trophozoite stage. So a trophozoite is the motile form, so you're going to look for motility. If the stool specimen is formed, we are going to look for the cyst stage, which is considered the non-motile form. You do need to preserve specimens. Oftentimes our hospital labs don't perform parasitology in-house anymore. So these are all going to be sent to reference labs for the most part. You can hold them at room temperature for 2 to 3 hours. You can hold in the refrigerator for 2 to 3 days. Again, if you know you get a specimen on a Friday and your courier has already come and gone, it's not going to go out till Monday. So you need to be able to prepare for that and know how to preserve that specimen. The other method, and typically the preferred way, is if you are doing a send-out, is to use an O&P kit. That has one vial that contains polyvinyl alcohol, or PVA, and the other tube is going to contain formalin. So depending on what we're expecting as far as parasites, based on patient history and sometimes patient travel, formalin will give us the better chance of recovering helminth eggs, larvae, protozoan, cyst, and then formalin can be used to make direct wet mounts and concentration procedures as well. The PVA is best for protozoan cyst in trophs or trophozoites. The PVA vial is used usually to do a permanent stain. If you only have a small amount of stool, you're going to put all of that into the PVA vial. When we're looking at stools for parasitology, you'll do a visual or a macroscopic observation. We're looking for bloody and mucus portions of the stool. Usually stool culture, as well as an O&P, will be 3 specimens, sometimes 2 to 3 different days apart, depending on how often the patient is going. Acceptance is going to be based on if the specimen and requisition match with the exact time and date of collection. So you have to make sure you're kind of mindful when you're looking at the specimen and requisition to make sure that they match. So your 3 general methods are direct wet mount, concentration procedures, or permanent stained slides. You can perform all 3 on soft and liquid stools. A form stool usually is only going to be a direct mount and a concentration. Soft stool could be a direct wet mount of concentration in the stain. A loose or watery stool could be a direct mount stain. You would expect trophozoites, so we don't necessarily need to do a concentration. Preserved stool we can do as a direct mount, concentration, and stain. So with the direct mount, you'll use a 2 by 3 inch slide, 2 preps of the same specimen, 1 stained and 1 unstained. Make sure that you dilute your specimens such that you can read newspaper print through the suspension. On the unstained slide, you're going to add a drop of saline with a small amount of specimen. On the stained slide, usually loose stool, you'll use a Quinzel stain or a Nair buffered methylene blue stain. If it's a foreign specimen, iodine also works well. So the picture that's up there is actually Giardia duodenalis. So you can see the trophozoite with the flagella, and that is going to be considered your motile form. And then you can also see the cyst form, and that's your non-motile form. You can see that's kind of rounded up, kind of looks like an egg. So after you get your suspension made, you're going to cover this with a 22 by 22 milliliter #1 thickness cover slip. The edges are either going to be covered with paraffin or petroleum jelly to kind of help seal it. You're going to read the entire unstained slide at 10x with the light down. If you see a suspected item, you need to then rotate into your 40x or your 100x and make sure that you're working from your scope with the micrometer, again, so you can measure. Then you're going to read the stain slide. If both the unstained and stained are negative, then you can proceed to the concentration and or if the permanent staining methodologies. So with the concentration method, this increases the density of all the diagnostic stages. Concentration does help eliminate fecal debris. One of the most challenging pieces of doing parasitology is determining what's fecal debris and what is actually a parasite. So the less you have to look through, the better. So the concentration method, the reagents that are used will also help preserve the morphologic features of the diagnostic stages. And we have two methods. One is a flotation, so everything comes to the top, so your diagnostic stages are floating free of debris or sedimentation. With sedimentation, you will either use xylene or ethyl xylene or ethyl acetate. Sedimentation is usually better than flotation, so we're going to have some centrifugation involved in this. And due to these chemicals and sometimes gas production in the stool sample, like I mentioned earlier, that's why you need that explosion-proof centrifuge, because nobody wants that exploding on them. Ethyl acetate sedimentation. So this is preferred when using fresh or preferred specimens. It efficiently removes debris while preserving diagnostic stages well. It generally provides better recovery of a wide variety of parasite eggs. For the flotation method, this is useful for recovering nematode and cestode eggs and protozoan cyst. It may cause distorted appearance of parasites due to the osmotic effects. It fails to recover operculated eggs, eggs with that lid-like structure, effectively. For sedimentation method, it recovers diagnostic stages from a larger variety of parasites than flotation, and it's better for detecting larger eggs and larvae. And this is an example of a permanent stained smear. Oftentimes we refer to these as a trichrome stain. So a Wheatley modification of a Gomorrah trichrome stain. So all your background debris will be a blue-green. Your protozoa or your white blood cells will have blue-green cytoplasm with purple or a reddish color. So what you're seeing on this picture is actually a trichrome stain. Since this is a permanent stain, we're going to start with 10x, but ultimately you're going to eventually go up to 100x, which is what this is showing here. Scotch tape prep. This is a different methodology. It is only used for Enterobius vermiculus, otherwise known as pinworm. So what happens is that the female worm will migrate to the anal area, usually early in the morning, and the eggs, or she will deposit the eggs around the anus. So pinworm is common in children. So before they use the bathroom in the morning, you can take Scotch tape and stick it to the skin of the anus. Then it slides into a little plastic tube that's then transported to the lab. And some of the tubes that they have like a plastic type of consistency, so you can lay that directly onto the microscope stage and read it from there. Note that if you have a light infection with Enterobius or pinworm, it may take up to 6 different specimens before you actually get a positive. And last but not least are blood samples. So things like Babesia, Plasmodium, Trypanosoma, those are organisms that are found in the bloodstream. And we will actually do what we call both thick and thin blood smears. So a thin blood smear is like your regular peripheral blood smear that you did in hematology. You're going to have to observe at 10x for a minimum of 30 minutes. These are very time consuming. Plasmodium and Babesia are oftentimes intracellular, whereas Trypanosoma are extracellular, so outside of the cell. A thick smear is a little bit different than your thin smear. So in fact, you're going to take about 3 to 4 drops of blood. So we're going to spread that because that's why it's thick. So instead of that 1 drop that you take in smear, this is actually, 4 drops, and you're not actually smearing it. It's going to kind of be there and continue to be thick. So you're going to start on your 10x, go to 100x for a minimum of 5 minutes or until you have observed 100 fields. And for some of our parasites, we do have serologic assays available, most of which are going to rely on the detection of antibodies produced to that parasite.